Nucleus. As we seen in the previous videos, we know that only eukaryotic cell will have the well-matured nucleus with nuclear membrane coverings. In plant cell, nucleus is located near the periphery, that is the corner of any one side of the cell, whereas in animal cell it is located near the center. It is why because in plant cell the size of the vacuoles are very larger that pushes the nucleus to one corner of the cell. We could see in the picture clearly. Whereas in animal cell, it is located near the center. Why? Because the vacuole size are smaller when compared to the plant cell. Nucleus is covered by a double layered nuclear membrane. It consists of chromatin fibers and one or more nucleolus. The nucleus also contains nucleopores, which we could see in the diagram. Nucleus is present in all living cells including plant cells except in matured mammalian RBC cells. It is because the RBC cells should need more space to carry oxygen to the cells. So that is why the nucleus is absent in matured mammalian RBC cells, whereas the nucleus is present in the premature stage of the RBC cells. Nucleus is considered as the center of control and place of genetic information. Why? Because as we said before, the chromatin fibers, which consists of chromosomes, which consist of DNA, that is responsible for the genetic informations. That is why the nucleus is the place of genetic information. The ribosomes, which are protein synthesis unit that are produced in the nucleolus. The chromatin reticulum is the site of genetic information, as we seen before, is normally present as fibers. Only during the cell division we could see it as a typical butterfly shaped chromatids. The RNA that are synthesized in the nucleolus carry informations from the chromosomes to the cytoplasm and also cytoplasm to the chromosomes. There are three types of RNA. They are messenger RNA, transfer RNA and ribosomal RNA, which are mRNA, tRNA and rRNA. Messenger RNA used to carry the message. Transfer RNA helps us to transfer this message from the cytoplasm to nucleoplasm or vice versa. Ribosomal RNA, which are responsible for protein synthesis, which supplies protein to the cells when they are needed. About the chromatin fibers, we could see in the upcoming videos of cell cycle. The next topic we are going to see is ribosomes. It was first observed by George Ballard in 1953. In the picture we could see the three types of RNA, that is mRNA, tRNA and ribosomal RNA. These three RNA are seem to be connected each other, which is possible by Mg2 plus ion for the cohesion of ribosomes. Here mRNA, messenger RNA, which is seems like connecting the ribosomal larger subunit and ribosomal smaller subunit. The amino acids are connected like a chain form, which is attached to one corner of tRNA. During protein synthesis, many ribosomes are attached to single mRNA and that it is called as polysomes or polyribosomes. Next we move on to chromosomes. In this picture we could clearly see that the chromatin fibers which are present inside the nucleus is made up of the chromosomes, and these chromosomes can be seen as a butterfly shaped chromatids only during the process of cell division, and these chromosomes consist of DNA, that is a double helix structure which has the genetic informations of the cell. Chromosomes. It was first reported by Strasburger. The word chromosome was introduced by Valdier in 1888. Bridges in 1916 was the first one to prove that chromosomes are the physical carriers of genes. Structure of chromosome. In this, we could see the primary construction which happened in the centromere and kinetochore. So the kinetochore consists of complex system of protein fibers which leads to the primary construction. Apart from primary constructions in the chromosomes are said to be the secondary constructions. We could see telomere at one end and satellite at the other end. Telomere not only prevents the fusion of chromosomal ends with one another but also offers stability to the chromosomes. It has important factor in determining the lifespan and reproductive capacity of the cell. It is also providing new sites into the conditions such as aging and cancer. Satellite chromosome or SAT chromosome, which is found in the other end of the chromosome. It is kind of identify marker, that is it could see only in particular chromosomes. For example, 13, 14, 15, 21 and 22 chromosomes will have this SAT chromosome. Based on the position of centromere, chromosomes are classified into four types. One is telocentric, second is acrocentric, third is submetacentric and fourth is metacentric. Telocentric is also called as terminal centromere, as one end we could see only the centromere, and terminal centromere capped by telomere is called as acrocentromere. And submetacentric, the chromosome have centromere as a subterminal, that is it is present, one side will have the larger arm and other side we have the smaller arm. And last is metacentric, we have equal sides of arms where centromere present at the median with the presence of satellite. Based on the function of chromosomes, it is divided into two types, autosomes and sex chromosomes. Autosomes are nothing but body chromosomes, sex chromosomes are nothing but allosomes. In human, we have diploid cell, that is we have 23 pair of chromosomes, whereas in first 22 chromosomes are said to be body chromosomes and last 23rd pair is said to be sex chromosomes which determine the sex of the individual.